Horizontal gene transfer has been very important throughout evolutionary history. We've seen horizontal gene transfer in chapter 21 when we talked about phylogeny and how we represent and study the relationship between living organisms. This tree of life shows the major domains. We've got bacteria, archaea, and eukaryotes. And the horizontal lines in color, the ones that are in blue, represent horizontal gene transfer across different groups of organisms. If you'll recall from chapter 21, we learned that vertical evolution is where parents pass their genetics to their offspring. So ancestral organisms pass their genetic information on to later organisms that are in their lineage. In horizontal gene transfer, genes are transferred from one species to another, or from two organisms of the same species that are not vertically related. So we can see here that this horizontal gene transfer indicates that there's been extensive sharing of genetic material across domains. We see sharing within kingdoms of bacteria, across domains between bacteria and archaea, between bacteria and eukaryotes, and between archaea and eukaryotes, or bacteria and eukaryotes. This one here indicates the transfer of the chloroplast. This one here is the transfer of the mitochondria, but you can see that there are other genes that have been transferred across and have been important in the evolution of each of these groups. Within bacteria themselves, it looks like about 17% of the genes in both E. coli and Salmonella have been acquired through horizontal gene transfer. And this is going to be pretty similar across a variety of different organisms. So not only are they related by common ancestry, but then they can potentially share genetic material with other organisms that are not closely related to them. Antibiotic resistance is an example of horizontal gene transfer that you are probably familiar with. In a healthcare setting, when individuals are heavily using antibiotics and antibiotics are prevalent, bacteria are prevalent in a healthcare setting, that antibiotic resistance may get selected for, and bacteria may transfer that antibiotic resistance through one of the three mechanisms of horizontal gene transfer that we'll talk about in just a few minutes. And so this, of course, can be a big deal because if you have a lot of resistant bacteria, they're very hard to treat, and then you end up with more bacterial infections. There are three types of gene transfer that we see between bacteria. So this is basically bacteria to bacteria horizontal gene transfer. There are two main ways that we see genetic diversity in bacteria. Bacteria are not performing sexual reproduction, so they're not combining genomes, they're not shuffling genomes, they're not gaining some of the diversity that we find in eukaryotes who are reproducing sexually. But because they're asexual clones of the parent cell, they are technically not acquiring any differences from the parent cell. So any differences that come up either have to come up by accident through DNA replication errors or through horizontal gene transfer, where they pick up genes from other organisms. If you take microbiology class, you're going to go through these in much more detail. We're just going to talk about the general ideas of each of these horizontal gene transfer methods. The first is conjugation. In conjugation, there is a direct physical interaction. In this case, it is a physical structure called the pilus that actually acts as a tube through which that genetic material is transferred from the donor to the recipient cell. So conjugation requires a physical contact between two bacteria that are near one another in their environment. In transformation, DNA is taken up from the environment. So in the lab, we do the P-glow expression of green fluorescent protein in bacteria, specifically in E. coli. And in that lab, you make the cell willing to take up the little tiny pea glow plasmid, and when it does, it acquires genetic characteristics that are different from the original cell. Now, in a real life situation, transformation can happen when a bacterium bursts or for some reason releases a plasmid, and a neighboring bacteria can pick it up from the environment and bring it into its cell. The last one is transduction. In transduction, a virus or a bacteriophage transfers genetic material from one organism to another. Now, this is not the viral DNA that's being transferred, it is the host DNA that is being transferred along with the viral DNA. Table 17.3 is a really great description of all of your gene transfers. You may have this one in your study guide packet. If not, you can find it in your online textbook. In the process of conjugation, the physical connection between two bacteria that allows for gene transfer, the donor cell must carry a specific gene or set of genes that are going to code for the production of that pilus or tube that is going to establish the connection between the two bacteria. That special plasmid is called a fertility factor, and it encodes all of the instructions for making the pilus, moving the DNA across, and then potentially even for replicating the DNA in the recipient cell so that it gets integrated into their genome, or at least maintained as a plasmid. Conjugation ability is relatively rare in a bacterial community. Only about 5% of E. coli strains in nature can act as donors. So a donor strain is the strain of bacteria that has this fertility factor plasmid. If they don't have that, then it is not a donor strain. It cannot donate DNA to the next cell over. If a strain does not have that fertility factor, it can acquire it. This would happen if that fertility factor is transferred through the pilus from one bacteria to another. In some cases, that fertility factor can be integrated into the genome as well. So when it is transferred over, it can be maintained as a plasmid, or it can be integrated, in which case it's a special case that is not going to get lost because it is encoded in the genomic DNA. If it's on the plasmid, there's a possibility the plasmid could be lost, no longer maintained by the cell. But as long as that fertility factor is present on a plasmid, the bacteria will maintain the ability to form the pilus. An F factor plasmid is going to carry all of the genes that are required for conjugation. They might also carry some advantageous genes that do something else for the bacteria. It doesn't have to just be the fertility factor. We say that a cell that has the fertility factor is called an F positive cell, and one that does not is called an F negative. So that F positive cell is going to be the donor. The F negative is going to be the recipient cell. So the cell that has that fertility factor is going to express the genes that are required to make a sex pilus. In this image here, we have a pilus that is growing, actually several, that are growing from this bacteria on the left out to the bacteria on the right. So our donor cell here is producing this pilus that is attached to the cell wall. It essentially punctures through the cell wall, and then genetic material can be transferred from one cell over to the other through that pilus. So once that fertility factor set of genes is expressed, it is going to produce a pilus from one bacterium to the other, and then that pilus is going to essentially pull it in like you would pull in an anchor on a boat, pull the other cell closer. And that's going to reduce the distance that that DNA has to travel across that sex pilus. Once that conjugation bridge is formed, the plasmid, or part of the genomic DNA, depending on if the F factor is located in the plasmid or in the genome, is going to be replicated and basically spooled across, unwound into the cell on the other side. So replication machinery is going to make a copy, send the copy through. Then in both cells, the plasmid is going to be copied so that it becomes double-stranded DNA again. So now we have a double-stranded plasmid in one cell and a double-stranded plasmid in another cell. Transfer of the strand of DNA is going to continue until the pilus is broken or until the DNA is completely transferred. If it is a small piece of DNA, the time of connection doesn't need to be very long. But depending on how long the transfer lasts, may or may not push all of the piece of DNA over. It really just depends on the size of the plasmid or the size of the genome DNA and how long that connection is made. After the transfer, the DNA can remain in plasmid form, be copied as a plasmid. It can also be integrated into the genome. If the gene that codes for the F factor is present and it is maintained in a plasmid, then the plasmid will continue to produce the pilus, or the genes on the plasmid will continue to produce the pilus. And then the donor cell will transfer that fertility factor over to the recipient cell, and the recipient cell will now be able to be a donor. Sometimes that gene, the fertility factor gene, is integrated into the genome. When this happens, this is a special situation called HFR, or high frequency of replication. This means that that fertility factor is going to be replicated every single time the cell divides. And it's going to be passed on to the next generation of cell when the cell divides. So it can be passed on in two ways. It can be passed on horizontally and vertically. Our second mechanism of horizontal gene transfer in bacteria is the process of transformation. Transformation does not require a direct contact between the cells. Instead, the bacterial cell pulls in DNA from the surrounding environment. Now, this is probably going to be present because when bacteria die, they lyse, they break, and their DNA is released. It can also be a plasmid that for some reason gets released. Just depends on what is sort of floating around the environment of the cell. Cells take up DNA by a special complex of enzymes that are on the cell surface, and that complex of enzymes has to be induced in order for the cell to take up DNA. So the cell's not just picking things up as it goes along. The conditions have to be correct in order for the cell to produce those surface receptors that are going to bring in that DNA. So in this example here, we've got a gene that is producing a special channel protein that is going to bring in a small piece of DNA, and then that DNA gets integrated into the genome. When we did the pGlow lab in class, you provided conditions for the reaction to make the E. coli cells that we were working with willing to take up the genetic information that you were trying to give it. This process is used extensively in molecular biology labs. It is the number one way that you get bacteria to take up the genes that you want them to take. So it's the number one way that you are going to create a transgenic organism. Transgenic just means that an organism has DNA from something else. So it is a relatively efficient way to get a gene into a bacterial system. The smaller the piece of DNA, the easier it's going to be to get it in. So a plasmid is going to be easier to get in than an entire chromosome, but a chunk of a chromosome might come in as well. In order for that piece of DNA to be expressed, it's got to be integrated into the genome. Unless for some reason it's an entire plasmid that can express the genes itself. If it's just a piece of DNA, it's got to be integrated to get in there. Transformation is not very common in the environment, although it can happen, again, depending on the conditions for uptake. For conjugation, the genes that get transferred are sort of based on chance and how long the connection has happened, how many genes get transferred across the conjugation bridge. In transformation, the rate of uptake really depends on the cell type and what special conditions there happen to be. Our last type of gene transfer is called transduction. Transduction is relatively rare, and the reason that it's rare is that it requires a virus to move genetic material from one bacterium to another. So this happens because sometimes errors occur when the virus is being packaged, the viral DNA, I should say, is being packaged into the capsid. If you recall from our lecture on viruses, we said that viruses not only make their own genetic material, but they also deactivate the host cell DNA. And they do that by cutting it into pieces. And so in the process of transduction, when the viral genome is being packaged, a piece of host DNA accidentally gets put into the viral package. So then when the virus infects a second cell and injects its DNA into the cell, it also transfers a gene from the previous host cell. So if the virus causes the cell to lyse, it may or may not matter. But if the cell does not lyse and instead integrates that gene into the genome, then that horizontal gene transfer has been successful and one bacteria has acquired another's genes through that viral carrier.